So we talked about some of the external structures that can be found in bacteria. So let's go ahead and start moving internally. And the first structure that we're going to talk about that's one of the internal structures is the plasma membrane. And all cells have a plasma membrane. It's very similar to our plasma membrane, in fact, in bacteria and archaea. So the plasma membrane is a phospholipid bilayer. It has, like we saw in the pictures before, 2 layers of phospholipids. They have that hydrophilic head and those hydrophobic tails that go on the inside. And the plasma membrane is what's responsible for keeping all of the internal contents inside the cell. So again, this is what that phospholipid bilayer looks like. You have these hydrophilic heads, and then you have these 2 like fatty acid or hydrophobic tails. And when you look at them, in their organization, you see we've got all these bilayers going on, but then we also have all of these like embedded proteins. And we have proteins that can be what we call peripheral, meaning they can be just on the inside or just on the outside. But we also have what are called integral proteins because they're integrated into the cell. And those integral proteins can sometimes just kind of transit through the membrane, or they can form pores or channels to allow the cell to transport materials in and out of the cell. So again, those peripheral proteins will be right there on the outside. Integral proteins are what we call transmembrane. They span the whole membrane. Now, the properties of the phospholipids give the membrane what we call some fluidity. So the membrane is actually not necessarily like a solid structure. It's actually pretty viscous. It's more of a thick liquid like olive oil, and it's self-sealing. So if there's a couple of small defects to the membrane, they can often fix that. Of course, if there are too many defects, then the cell can't fix it. But the fluidity, that viscosity of the membrane allows both the phospholipids and the proteins within the membrane to move. And these proteins can move in order to function. Sometimes proteins need to come together in order to do some sort of certain function. And these phospholipids can also move. They can rotate, they can move over here. So it's not a static or still system. It's a system that moves. And the function of the function of the plasma membrane specifically is, like I said, to keep the things inside the cell that need to be in the cell. So it's a container. But it also can serve as what we call a selective barrier. The plasma membrane itself, not considering the proteins or anything like that, the plasma membrane itself only lets certain things in. They usually have to be very, very small. And it can also depend on the charge or the polarity of the molecule. Oftentimes, cells don't necessarily want to let things in that are like ions. We have to use specific transporters to get ions in. But sometimes water can freely flow through the membrane because it has a little bit of polarity that allows it to come through. Now, this is going to be unique to the prokaryotes. The enzymes for generating ATP, the electron transport chain enzymes, are found in the plasma membrane. In eukaryotic cells, those enzymes for ATP production, that electron transport chain, is found in the membrane of the mitochondria. So because prokaryotic cells don't have a mitochondria, they have to put their enzymes somewhere, so they put them in the plasma membrane. Now, when we're talking about photosynthetic prokaryotes, in eukaryotes, the enzymes responsible for photosynthesis are contained in the membranes of the chloroplasts. Remember, we don't have chloroplasts. So there can be these kind of infoldings of the plasma membrane where the photosynthetic pigments and the enzymes for photosynthesis can be found. We can also have damage to the plasma membrane, and like I said, sometimes that damage can be self-sealing, the cell can fix itself, but sometimes that damage can be very severe and lead to cell death. And some of these compounds here, like alcohols, detergents, and certain antibiotics like polymyxin, directly affect the cell membrane. The immune system can also directly alter the cell membrane. So we call it the complement system. It's a cascade of protein reactions that can occur because of the immune system, and that also damages the plasma membrane. When the plasma membrane is damaged, it no longer functions as that container to hold the cellular contents in, and so you get leakage of the cell contents. And that causes cell death. So it's very important for a cell, regardless of whether or not it's a prokaryote or eukaryote, it's very important for those cells to retain the integrity of their membranes. When we talk about that second feature of the plasma membrane, that selective permeability, there are a handful of different ways that compounds or elements or needed materials can make their way across the membrane. There are passive processes. And when we say passive, we mean it does not require the expenditure of ATP. It's free, right? You don't have to pay anything. And there are two major types of passive movement across the membrane. The first is called simple diffusion, and the second is called facilitated diffusion. With simple diffusion, what we're looking at is that outside the cell, there's a high concentration of something we want or need. Inside the cell, there's a lower concentration. Depending on the characteristics of the molecule, as long as it's small enough, it's not charged, it's got the right polarity, these molecules that are on the outside will diffuse. They basically will freely move through the plasma membrane until the concentration is the same on the inside and the outside. And that does not require energy because the molecules on the outside are following the concentration gradient to the inside. If there were a higher concentration of molecules on the inside, they could then diffuse to the outside. So it can go both ways. It just will always go from an area of high concentration to low concentration. With facilitated diffusion, we're also looking at this idea of having a concentration gradient. But this time the molecule might be something like an ion that's charged, or it could be large like glucose. So we need proteins to help. Now these proteins can be kind of a non-specific, like a pore, where the ion can go in the pore, or they can be a little bit more specific where it's like the glucose transporter, so the glucose can get in and then be used inside the cell. But again, this also works with the concentration gradient. There are more ions on the outside, so ions can come in. There's more glucose on the outside, so glucose can come in. It doesn't require any energy because they're moving with the gradient. Water can also move through channels, but it has to be from the high to the low concentration. It can move two ways. The first way is the one I talked about before, where water can actually get through the membrane. Okay, it can. Water can also go through channels called aquaporins. So you have these water molecules, and they can go in. Water also moves with a concentration gradient. It moves from an area where there are fewer solutes, meaning there's more water, into an area that has lower water. So they will move with that gradient. And this is what one of those channels looks like. So individual water molecules can go through the membrane, but they can also be transported maybe faster through these types of proteins called aquaporins. They're pores that transport water. Now, when we're talking about water and the cell, there are some different things that can happen. So water is responding really to the concentration of solutes. And so if the concentration of solutes is equal inside and outside of the cell, water's moving in, but water's also moving out, so the cell kind of stays the same. If there are more solutes inside the cell than outside, the water tries to go into the cell to kind of dilute out what's in the cell. So this is called a hypotonic solution. There are fewer, that's what hypo means, fewer solute molecules outside than there are inside. So the water tries to go inside to dilute those solute molecules out. When a cell has a very, very, very strong membrane, or if the cell has a good cell wall, they can tolerate this for a while because the cell wall or the very strong membrane can hold it together. However, at some point, the cell wall or the membrane might give out, and that can cause the cell to burst. And we call that osmotic lysis, where the water rushing into the cell causes the cell to burst. Now, in an opposite situation where there are lots of solutes outside of the cell and very few on the inside of the cell, we call that hypertonic. There's more on the outside. So in this case, what will happen is water will rush out of the cell to try to dilute the outside, and that shrinks the cytoplasm and can dehydrate the cell. So that's called plasmolysis. It's not really a lysis because the cell's not breaking, but that's what they call it. So it's important for cells to be in solutions that are isotonic for them. Sometimes we do like to have plants in a slightly hypotonic solution because it causes the cell to be more rigid. So we like that rigidity in plants, and that's why plants might start to droop if they don't have enough water because they can't put the water into the cells. So you give them more water and they'll perk back up. And this is the principle behind using things like salt to preserve meat. Not because we're worried about the meat cells, but because you're worried about the bacteria that will break down the cells. And so by putting salt or sometimes even sugar, you dehydrate the bacteria because they lose their water to dilute that compound. So what happens in conditions where perhaps you want to move something against a concentration gradient? Let's say you have sugar inside your cell, but you want more. You want more sugar. And since you already have more inside your cell than outside, you have to go against the gradient. When you're going against the gradient, we call that active movement, and it requires energy. It also does require proteins. So there is nothing that can move against the concentration gradient directly through the membrane. You have to have proteins to help do that. So with active transport, you literally just use ATP to open up the transporter and bring stuff in. It's the most simple type of active transport. In a second type, you use what's called group translocation. So let's say you want to bring glucose in against the concentration gradient. You add maybe a phosphate group. And what happens here is maybe the phosphate group is going with its gradient, but the glucose is going against. So because the cell wants to bring in the phosphate, it will also bring the glucose. And you might be saying, well, where did you use the energy then? You said active requires energy. I did. I said active requires energy. It requires energy to add and modify the molecule you want to bring in. So in this case, to add the phosphate group, it costs energy. So then the actual transportation itself was free, but you had to pay for the product. It's like when you buy something and they give you free shipping. It's not really free shipping. It's built into the cost of the item. So you wanted to bring in the item, you paid. And then it shipped for free. So what is this magical liquid that we're trying to protect and move solutes in and out? Well, it's the cytoplasm, the substance inside the plasma membrane. The cytosol itself refers to the liquid component, and that's about 80% water. There are lots of proteins, carbohydrates, lipids, ions, and of course, cytoskeletal components that can make up the rest of that cytoplasm. And with the cytoskeleton, a lot of times people don't think about bacteria having a cytoskeleton. We think of that as being a more eukaryotic thing. But cytoskeletal elements do exist within prokaryotic systems, and they have homologs in eukaryotic systems. So these cytoskeletal components are so important that they've been preserved from bacteria all the way up to eukaryotes. So bacteria have a homolog for our protein called tubulin, and bacteria use it as part of their process for cell division, like to split off cells. Bacteria have a homolog of actin that they can also use to help facilitate cell division. And they have intermediate filaments, or a homolog of intermediate filaments that they can use for cell shape. And so to get into a little bit more detail, the bacterial protein here, FtsZ, recruits other proteins to the division site, and it forms this ring that causes the cells to split during cell division, and it's a homolog of tubulin. In cells that lack this protein, they can't divide, and so you end up with these really long filaments of multiple cells. So this protein is critically important for them to be able to divide appropriately. This MREB is a homolog of actin. Normally in the cell, it's found kind of distributed throughout the whole membrane, the whole length of the cell as these kind of bands that surround the cell. During the cell division, they come over here to the middle, and they also are part of this process of the cell splitting into two. If there are mutants in this protein, instead of, like, if it's a rod-shaped cell that's supposed to have this, that helps like all kind of along the whole length of the cell, mutants are round. They don't hold their cell shape appropriately. And then finally, there's this protein called CRES. It's similar to intermediate filaments. In this particular species called Caulobacter, we're trying to illustrate here that they're curve-shaped a little bit. If they don't have this protein, then they're a total rod. They lose the curve that makes up their cell shape. So again, bacterial proteins can make up part of the cytoskeleton, just like we see in eukaryotic systems. And bacteria also can have their own unique cytoskeletal proteins. They have proteins that belong to this family called MIN, M-I-N, that helps to determine when and where the septum will form. The septum is the part that helps with the cell division. There are also PAR proteins, P-A-R, that are involved in helping a cell determine where to keep its plasmids.